Welcome students. Today we are going to learn about isolation techniques for isolating Rhizobium species and Frankia species from the soil. The objective for this would be to know about these two bacteria, understanding their characteristics, how they grow, and to learn about special techniques for isolating these from the soil. The Rhizobium species are bacteria and they are present in soil. Predominantly, you will see them growing in the region near the root. The roots of various plants are attacked by many bacteria, but Rhizobium prefers leguminous plants like clover, peas, beans, like this. Rhizobium bacteria will be present in the soil away from the root. You will find them in close vicinity with the root, and you will also find them very well attached to the root hairs. Now this is a region from where they will enter into the roots and cause the formation of nodules in the leguminous plants, where they will actually bring about nitrogen fixation. This is the most important act they do in the symbiotic association with these plants.
Ma'am, why is it so that the number of rhizobium present in the rhizosphere layer is more than the number of rhizobium present in the soil?
That happens because you see the plant will produce certain extraneous material like some excreted material, some secretory products, and all those coming from the root would be present near the region of the root, that is the rhizosphere region. So these rhizobium gets attracted towards these by a chemoattractive method, and they remain very much associated in close contact with the roots, and the numbers are that's why high. This picture shows rhizobium cells within the root hair cells. They are gram-negative, pink coloration, and they are motile and non-sporulating rods. This is a typical example of a root with infected rhizobium giving rise to nodules. You can see the large structures called as nodules on the typical plant Trifolium subterraneum. Initially, a lot of work had been done on nitrogen fixation being brought about by plant species. Nobody knew that it was microbes which were in symbiotic association with plants which was doing all this. It was M.W. Behring who in 1888 actually found out a bacterium, named it Bacillus radicicola, and proved that this was in symbiotic association with legume plants and was doing nitrogen fixation. In 1889, the first species of rhizobium, that is Rhizobium leguminosarum, was identified. This is a classification protocol for the typical organism Rhizobium. It belongs to the kingdom Bacteria, phylum Proteobacteria, class Alpha Proteobacteria, order Rhizobiales, and belongs to family Rhizobiaceae. This you can see Rhizobium meliloti on mannitol salt agar, which produces watery white colonies. Now we have seen briefly about Rhizobium. We will now learn about other nitrogen fixing bacteria, because Rhizobium is not the only one. There are many others. Now we know that nitrogen is present in the atmosphere in a huge amount, approximately 78%, but cannot be taken directly by the plants. Nitrogen needs to be converted to ammonia or nitrate for incorporation into plants. And there are several categories of microbes which does this work. In the nature, lightning does some of it, the percentage is very low. And in the industries, we have the production of ammonia in a typical chemical process requiring a lot of catalyst, pressure and temperature, which produces ammonia. Otherwise, nitrogen is very difficult to be converted to either nitrate or ammonia, because it has a triple bond which needs a lot of energy to be broken down and then combined with hydrogen or oxygen. So this wonderful work is done by the nitrogen fixers, and they can be categorized as either symbiotic microorganisms, asymbiotic or free living microorganisms, and some grow in association form. We have a list of several organisms here, some in the symbiotic category, meaning they will be associated with a typical host plant. There are some free living which remains in the soil. And then we also have some associative growth. Typically, like example, azospirillum grows in association with grasses. But for our protocol experiment, we have chosen the Rhizobium legume symbiosis, the typical bacterium Rhizobium, and we have used the Frankia species. So far, we have seen about Rhizobium. Now, we will briefly see about the Frankia. Frankia are actinomycetes. They are slightly different from typical bacteria. They produce mycelial-like structures and they differ on their colony morphology as well as microscopic view from ordinary bacteria. Here you can see an association, symbiotic association of Frankia with the alnus producing root nodules. It was first Albert Berhard Frank who in the 1800s discovered the Frankia species, and the genus Frankia is named after him. Frankia generally colonizes these two species, that is the alnus, commonly called as alder trees, and the casuarina species. The alder trees are present in the typical temperate forests, while the casuarina you will find in the subtropical and tropical coastal belts. We have already understood that Rhizobium remains present in the nodules as well as in soil. Isolation of Rhizobium from nodules is easier and often gives better results, yet we are moving on with our experiment to see how we can isolate these from the soil.
Why is it so that it is easier to isolate Rhizobium from the nodules than from the soil?
Because, see, the nodules are very specifically housing the Rhizobium within itself. While if you are trying to take soil from around this region, that will be also having some other contaminating microorganisms. So you will have to eliminate the contaminating microorganisms, go for several rounds of pure culturing probably, while in the nodules it's only the Rhizobium. So isolation and identification both becomes very, very easy. This is a typical growth on East mannitol agar medium, but we have incorporated the medium with Congo red, a dye. This helps us in identification. If you are doing it from soil, there will be several other organisms, very commonly agrobacterium species. Now you can differentiate because Congo red is absorbed by the Rhizobium cells only faintly, while most other bacteria will absorb it very strongly. So if you find very light pinkish or whitish color colonies on a plate like this, they are of Rhizobium. While if you have dark pink stained colonies or reddish colored colonies, those are of other organisms and not Rhizobium. The requirements are the medium East mannitol agar can be supplemented with Congo red and the soil sample taken is rhizosphere soil. There are other requirements like petri dishes, conical flasks, culture tubes, you need sterile water, sterile pipettes, and likewise many other requirements like the cotton and other things, filter paper that is there in a microbiology lab. First, we will prepare the medium. This is the composition of medium where we have mannitol, dipotassium hydrogen phosphate, yeast extract, magnesium sulfate and NaCl. Obviously agar because we are using a solid medium. We will be measuring these in the typical composition that is mentioned here, then dispense it into a flask, make it up to 1 liter and then autoclave for 15 minutes at 15 pounds per square inch pressure and then cool it down. Then we will be actually also doing the soil collection during this time because this is a very important part here. Collection of soil requires sterile containers, sterile augers. If at all it is not sterile, as far aseptic processes as possible should be taken. Now we are collecting rhizosphere soil, but we will be taking some 3-4 samples and then mixing them together. While we bring it to the lab, if there is a time lapse, it can be stored in the refrigerator. After the soil collection, we take the soil sample, mix it thoroughly, weigh 1 gram from it and go for serial dilution technique. Now obviously serial dilution technique is a very important and required frequently in the microbiology laboratory. Aim of this would be to dilute the sample as far as possible and then see if we can isolate discrete or individual organisms from a typical sample. So here we can take 1 gram of this sample and take 9 ml broth or sterile distilled water would make it a 10 power minus 1 dilution, meaning it is 1 by 10 times diluted. So now if we take 1 ml from this and mix it into the second tube containing same 9 ml, then it will also give another fold dilution resulting in 10 power minus 2 dilution. Likewise, we can go from one tube to the next, making the sample more and more diluted. We can carry it out till 10 to the power minus 6 or 10 to the power minus 7. So this is a serial dilution protocol which needs to be followed with the soil sample. After the serial dilution technique, we will follow a plating technique. We will be taking melted agar in tubes and then pouring it into the petri dishes. The petri dishes taken should be sterile. Take 0.5 ml of samples from each of these dilutions that you have made and add into petri plates. Then it is rotated clockwise and anti-clockwise so that the sample mixes well, and then it is allowed to go into the incubator where it is kept at a constant temperature around 20 to 25 degrees centigrade. Rhizobium grows very well at this temperature, can be kept for 2 days or 48 hours. The plate should be inverted while you are keeping it in the incubator. Then once you have waited till the 48 hours, you will observe for growth on the petri plates. If you take yeast to extract mannitol medium, you will see watery colonies like this or white colonies. If you take the Congo red medium, you will also see some light pinkish colonies which are obviously of Rhizobium species. If there are other organisms present, they will be darker in color. So once you know which organisms to pick from these plates, then we will be carrying out a streak plate method, subculturing method for getting a pure culture. Here we are trying to obtain a pure culture of the organism Rhizobium from the initial isolation. Here you can see the watery colonies. Then we perform a gram stain. So we have performed and we have got gram stain showing gram negative rods, which obviously are conclusive of some Rhizobium. But we cannot conclude that these are Rhizobium only by gram staining technique. We will also have to carry out some other tests. We carry out a capsule staining and we'll find that no capsules are present because the Rhizobiums are not capsulated. Yet there is a staining structure you can see around the cell which takes the stain. This is not of a capsule or of a slime, but mostly of some polysaccharides or gummy substances which are produced by the bacterium. This is also true because Rhizobium produces huge amount of extra polysaccharide material. Then next the two tests are performed, that is catalase and oxidase. What do we do in catalase? We take 3% hydrogen peroxide solution onto a slide and then add onto the culture of the organism that we have grown on the plate. If there is effervescence, you can see here bubbling, that is true of a true positive reaction, or that means the catalase enzyme is present. If there are no bubbles, that means catalase is absent. Likewise, we also carry out an oxidase test. We take the typical reagent NNNN tetramethyl paraphenylene diamine dihydrochloride. This reagent, when added to oxidase strips, will and the organism touched upon it, will cause a purple coloration or a dark bluish coloration if the organism produce oxidase. If the organism do not produce oxidase, you will have no such coloration. That means the test is negative. In our case, if we are trying to isolate Rhizobium, they should give positive catalase test and they should give positive oxidase test. We also carry out another experiment by the name hanging drop technique, which shows whether the organism is motile or not. There is a typical slide called as a cavity slide, and what you do is you take the suspension of the culture onto a cover slip first. You put it on the cover slip and then invert the cover slip onto the cavity of a cavity slide, and then you focus it under the microscope. If the organism is motile, you will find that just directly without any stain under the microscope. Rhizobium species are motile because they have flagella by this simple protocol, and it will be easier to identify the rhizobium. Next, the tests that are carried out are the dextrose fermentation tests. Here, glucose or dextrose is taken in the medium and the organism is inoculated. We also incorporate a pH indicator like phenol red. If the organism ferments glucose, then the medium has acidic conversion and the indicator that is incorporated changes color to yellow. It proves that the organism can ferment dextrose. And we know that rhizobium can ferment dextrose, which is very evident from the tubes here. You can see that it has turned yellow, and Durham's tubes, the presence of bubble, that shows gas has been produced, which shows that rhizobium species may be present because glucose has been fermented to acid and gas. So with those results, we actually come to a conclusion that obviously rhizobium could have been isolated from the soil. Basically, rhizobium is a nitrogen fixer. Agriculturists can go for the production of biofertilizer in this way. They can isolate rhizobium either from root nodules or from soil and can grow them in large scale or in huge amounts, can supply them to the agriculturists in the form of biofertilizers while they can use it in the field. That will enhance the nitrogen take-up ability of the plants because rhizobium will be fixing the nitrogen there. It is actually very difficult to isolate Frankia species from soil because they are very closely seen associated with their symbiotic partners, the alder and the casuarina, and you will not find much of them in the rhizosphere or the other region. But we will still try, and we will have specific medium for isolating these organisms. Q-mod medium is the choice of medium which contains several salts. There are minor salts and even lipid supplements which are needed for Frankia growth. They are fastidious and they actually do not grow so well on media. They are actinomycetes, so the generation time is also very high. It is approximately 15 hours or so and requires 2 months for proper growth and isolation. This you can see the growth of Frankia on alder on a typical Q-mod agar. Because they are actinomycetes, you will find the growth of the colony centrally housed and anchoring close to the agar and the filamentous like growth present on the other surface. Here you can see a microscopic observation of Frankia alni, and the long branch structures are the mycelium of Actinomycetes frankia. These are branched septate hyphae, while the vesicles that you can see are thick-walled cells. They are called as diazovesicles with continuation with the term diazotrophs, meaning nitrogen fixers, because this is the region where nitrogen is getting fixed. Nitrogen fixation is brought about by an enzyme nitrogenase which is oxygen sensitive. So, most organisms which fix nitrogen generally have some or the other means by which they can exclude oxygen from the nitrogenase enzyme. In this case, the Frankia have the diazovesicles which you can see here, where they are compartmentalizing and not allowing much of oxygen diffusion in these areas so that nitrogen fixation can be done.
Ma'am, you said that nitrogenase enzyme is oxygen sensitive. Then how does rhizobium protect this enzyme from oxygen activity?
In case of rhizobium, in the nodulation process, they are fixing nitrogen. They are not doing so when they are in the soil. Okay, so in the presence of a plant host where they are producing nodules, they get the help of the plant to produce a specific compound called as leg hemoglobin. It is very similar to hemoglobin which carries oxygen. In case of rhizobium, the leg hemoglobin stands for leg for leguminous. So in the case of leguminous association, the rhizobium will produce this leg hemoglobin, which will help as a carrier or binding of oxygen and will not allow much oxygen to diffuse into the differential structure produced by the rhizobia after they have entered the root cell and transformed itself into bacteroids ready for nitrogen fixation. So the leg hemoglobin does the work there. There is no such compartmental structure like this, but leg hemoglobin in the bacteroid structure will help in the regulation of oxygen diffusion into the bacteroid so nitrogenase can be protected. So nitrogenase actually is the enzyme which does all the work of nitrogen fixation, which we can see here in case of Frankia. These are the typical compartmental structures which houses the nitrogenase enzyme, which converts the nitrogen which cannot be easily taken into ammonia, and then that can be incorporated into proteins and other structures containing nitrogen in the cells. Now, two cultures you can see the growth characteristics of the Frankia species. They are very much oxygen sensitive as well. You can consider them as microaerophilic or requires less oxygen. So when you are growing them in the conical flask in broth, they should be not agitated or shaken. If you agitate or shake them, you will see pellet formation because they are mycelial in structure, while if you just leave them static, they will have the diffused growth. In case of Petri plates, when you are growing them in medium, taken on Petri plates, Petri plate lid should be covered with petroleum jelly. Now, for isolation of this organism, we generally follow a similar protocol of serial dilution, pore plating as we did for rhizobium. Very similar approaches are done. The difference is with the medium, we have a separate medium, and in the incubator, they are kept at very low temperatures of 20 degrees and should be kept for 2 months. It takes longer for the growth of Frankia. As already mentioned, they are actinomycetes. The generation time is very large. Two other problems are associated with isolation of Frankia. The first one is most often there is a contamination problem. Even if you isolate from nodules, they are associated with so many other surface contaminants that when you grow them on medium, the fast growing contaminants grows fast, while the Frankia being slow growers cannot grow. So obviously you should plate into several Petri plates or several conical flasks, and if there is growth before 2 months, you should discard all those, understanding that there probably are contaminants. Frankia cannot be very easily isolated from such contaminating plates or flasks. So this is a simple protocol, and for identification we should go for gram staining. In gram staining, Frankia will appear as long branch rods, and in media they will have a colony appearance, which already has been discussed. And not much work has been done with Frankia because although they are nitrogen fixers, there is less chance of these being used as biofertilizers. The time taken will be so much that your cost will not come out. So biofertilizer from Frankia is very less produced. We are more using rhizobium and several other cyanobacteria for such biofertilizer processes. But understanding about Frankia and rhizobium has already helped us understand that nitrogen fixation process will obviously help in the agriculture. We have understood the isolation techniques for rhizobium and Frankia. We have seen the typical composition of some media required for such isolation, how to go for soil sampling techniques, serial dilution protocols, which will help us understand how you can grow them and isolate them from simple substance like soil and go for cheaper biofertilizer production.